Well, thank you. Thank you very much for having me. Originally, I was intending to be on campus. I'm glad that kind of coordination did not work because we had COVID for the first time in my whole family, like a couple weeks ago. That was insanity. So we're just now barely recovering. So I wouldn't have been able to realistically travel anyway. So, I'm so glad to be here to tell you a little bit about the work I was doing, what I've been doing, and how it might relate to the theme of the conference. And I'd way rather be in Marseille hanging out with you guys. So I'm sorry to miss you. So my lab has been focused for the last 20 plus years on this idea that every patient deserves their own equation, which is sort of the overall rubric and overall metric for ways of thinking about metricizing elements of patients' care so that you can integrate it together to sort of personalize and optimize care. And the theme of this meeting obviously has to do with sort of evolutionary biology approaches and how you track the evolving disease or understand the evolving disease. And so I'm going to tell you a couple vignettes and connect them together through the lens of understanding sex differences, understanding heterogeneity in drug responses or therapeutic responses, and radiomics, which are image-based modeling. So I'm currently at the Mayo Clinic in Arizona, and I'm focusing mostly on a type of brain cancer known as glioma or glioblastoma. Glioblastoma is the most aggressive of the gliomas. It's really the most deadly of the brain cancers in general. It's not only a deadly cancer, but it's a neurological disease in the sense that you'll see in the following slides, in the sense that these tumors are diffusely invasive into the brain. So the cells are in constant communication with the brain as well as with themselves, the tumor cells themselves. So there's a complex dialogue that is become increasingly more clear, that complexity of that dialogue over the last several years, including the work by Michelle Manji and a variety of other labs who have focused on the integration of the brain, of the tumor cells with the actual brain networks themselves. So it's crazy science. There's really lots of interesting biology to peel back. So the incidence of this disease roughly in the US at least is roughly about 13,000 cases. So it's not a breast cancer, thankfully, because it's an unbelievably aggressive disease. The current standard treatment is surgical resection to the maximal possible extent while retaining function, followed by radiation therapy for 6 weeks, followed by and during that radiation therapy, patients receive a certain kind of chemotherapy known as temozolomide, and followed by adjuvant chemotherapy. And that usually is about that will get patients through, 6 to 8, 10, 12 months before they recur. And ultimately, the patients succumb to their disease, at about 16 months on average, with 5-year survival only being at a very generously 5%. There hasn't been a lot of new drugs or treatments approved over the last 30 years. So basically, we're running a lot of clinical trials, but we're not really moving the needle. And so what explains our lack of moving the needle? Well, every patient deserves their own equation because every patient is different. So here's 15 newly diagnosed glioblastoma patients with the same treatment, receiving the same treatment. You'll see that we've scaled the tumor size on the y-axis to be 1, to all start at the same. And all the patients are only receiving biopsies only, so they're in eloquent locations in the brain for which surgical resection was not possible. And I did that so that you could see the relative growth dynamic without like a sudden change having to do with surgery. And you can see that there's, even with the same radiation and chemotherapy window and adjuvant chemotherapy, there's just a huge diversity across these patients, such that if this is the standard of care, which it is, then patients like this blue patient at the top of the picture is probably not receiving much bang for their buck from this therapy. Whereas this patient down here, this orange patient, is receiving a huge benefit from this therapy. And these patients on the bottom are probably the ones that were slowly improving these trials of the last 50, 40 years. Again, the needle does not move quickly in this disease. So I mentioned the complexity of this disease, and I think it's best visualized with this sort of picture. So if you've got the brain in the background and you've got the abnormality that you see on MRI segmented in red. And if you were to zoom in at a single voxel level, you would see exactly this. These are tumor cells migrating and invading. Black is the normal brain. That's where the neurons are, the astrocytes are. There's a mix of neurons and astrocytes in where the red area is as well, because the tumor cells are not completely evacuating the space of all other things. They're still communicating with those cells, But way out the periphery, you can think of it as just sort of normal brain where there's these single individual cells pioneering out. So as a mathematician, I look at that, of course, and I'm like, okay, well, this is a complex system of interacting agents, and we might have some rules about this system, and let's see if we can figure out what those rules are and figure out how to predictively model what's going on with this disease. So a huge component of my lab, which I'm actually not going to focus on today, was focused on this. I'll talk about a few aspects of this sort of predictive modeling. So if you do individual cell tracking of these tumor cells, what you find is, in fact, that there are diffusion is a reasonable estimator of the invasion process. And then we have a proliferative dynamic, and we have some sort of response to therapy, and you can build reaction diffusion type models that explain this sort of what you see under the microscope here. And what we've done is we've built a variety of tools that allow us to take patient MRI images, what you see on the top here, to predict the model parameters and personalize the model parameters for each patient. And then that allows you to generate the predictions on the bottom of diffusely invaded tumor. So again, what you see clinically is the image on the top, and what the actual reality, because of that diffuse invasion you were seeing in the previous slide, is the picture you see on the bottom. Red being low cell density, few tumor cells that, a few red cells way out in the normal brain, and white being frank tumor, basically mostly tumor cells at the core, and highly, clearly identifiable even with the naked eye. So using tools like the reaction diffusion equations I just showed you in the previous slide, and personalization techniques we built to be able to estimate those model parameters, we can do one aspect of predictive understanding of at least sort of the general trends about what's going to happen for the patient's disease. But as I promised in this presentation, this talk was going to be about sex, drugs, and radiomics. So let's first talk about sex. So there are sex differences in the incidence and outcomes for GBM. And this data has become increasingly clear since the Stoop protocol, the protocol that I just laid out to you, became more standard of care. So although there's a small subtle difference in the SEER data, which is a population level reporting outcomes database for understanding incidence and epidemiology, basically, of how often do we see gliomas or glioblastomas in the community, you see that there is a separation forming between the male and female lines in terms of outcomes. And there's a significant sex skew such that there's more males than females that have this disease, 1.6 to 1 ratio. So there's lots of data sets I can show you. I can show you our own internal data set, which looks strikingly like the Ohio data set. Same story. So we became interested as we were studying those previous models, those reaction diffusion models, where you were tracking tumors over time and building these reaction diffusion mathematical models for predicting the dynamics of the disease. We built a very large database. So we have a large database going on 40 some odd hundred patients that we followed serially over their lifespan. And we've got, something like 100,000 MRIs for those patients. So we have a lot of data to be able to feed those sort of reaction diffusion models, but it also allows you to think about the data set differently, because it becomes a data science sort of question. And so I'm going to show you some of the statistical and more data science side of things that we recently worked on. So in this interest in sex differences, we partnered with a colleague who really introduced me to the idea that there should be sex differences in these data. This gentleman, Josh Rubin, his lab is at Washington University in St. Louis. And we've started looking at integrating transcriptomics, imaging, and survival and outcome data. And the first thing we did was some jive analysis, which is joint and individual variants explained. So this was run by a statistician, Rosie Liu. You see the very second to last author on the paper. And she focused on this method, not to say, let's analyze males and females separately, because that does lead you to some signal, but you expect there to be a significant common signal between males and females. And the difference is biologically, we would hope, would be, sort of more marginal, right? And so to elucidate that marginality, we used what's called the jive analysis with joint and individual variants explained. And what you do is you take the male data and you take the female data, and you combine them together and you ask what's common between them. And that's the joint variation. That's this joint graphic here on both of them. And then you have a residual that's basically a female and a male specific component. And then you have this residual noise, right? So what you then look at is the female specific and the male specific component. What is that signal? How is that signal different than the joint baseline? And what you find is there's a significant number of distinct genes that are expressed and patterns thereof. And if you just do a clustering analysis, which we kind of see in these pictures, there's about 5 different clusters on both sides. There's 5 female-specific clusters and 5 male-specific clusters as a first pass of something to do with this sort of data. You then look at those patients, then look at those data, and you ask the question, okay, what's the most interesting cluster on either side, right? So let's look at the female-specific. The most interesting cluster is the long-lived patients, let's say. So what biology describes long-lived? And you find that it's integrin signaling is the biggest biological signal in the long-lived cluster of patients. And then you do the same thing on the male side, and it's cell cycle control. So there's different biological mechanisms that are leading to long survival in these patients. So that is the first finding. The second was, given this information, could we then go look at the patient imaging data and say, well, integrin signaling suggests something to do with the diffuse invasion pattern, potentially an invasion question, whereas cell cycle control is more like proliferation control, proliferation dynamics sort of questions. And that's exactly what our reaction diffusion models were working on, right? On estimating. So we summarize here a synthesis down of hundreds of patients of data. I don't have the N on this slide, but I think it's 500 patients, 509 patients or something like that. We looked at on this particular slide, where we looked at either overall survival, or we looked at the subcohorts of patients that have extreme survival, meaning they lived greater than 5 years, or short-term survival, which was the subcohort of patients that lived like less than 6 months. And we asked, what is the male correlates for, let's say, overall survival, and the imaging features, which I show in gray, and age, which is the #1 most common predictor of survival. And we find that the males certainly correlate, their overall survival definitely is in Cox proportional hazard analysis, definitely relates to age, but it also relates to tumor size, T1 GAD weighted MRI radius on MRI, whereas the female overall survival correlates with age and diffuse invasion patterns, which is kind of consistent with that integrin signaling story. You can look at this objectively, again, for the short term and the extreme survival, but the whole point to this image is not the exact details. The point to the image is the male and female patterns are different, is the point. So what you saw on the previous slide was the transcriptome. Transcriptomics describing long-lived patients is distinct between the males and females. And so basically what describes what biology leads to long-lived patients is different between males and females, and what imaging features that might reveal aspects of biology connect to our overall outcome are different between patients. So everything I've shown you over here so far has really been kind of patient level, right? We've talked about a very homogeneous diffuse invasion of tumor cells, where there's this one rate associated with diffuse invasion and one rate associated with proliferation, and sort of is like a homogeneous single characteristic of the overall tumor. But the tumors are way more complex than that. So there is a ton of intratumoral heterogeneity, and GBMs glioblastomas, GBMs are known for this intratumoral heterogeneity. So if you look at, I like to sort of analogize this to MRI being a satellite map onto the brain. And so let's do a satellite map onto a country. So we'll pick the United States, and you biopsy in some location and you get an answer. So you only get one little biopsy, right? And that set of biology, whatever's represented in that piece of tissue, is the driver for the clinical decision, right? If there's some gene broken in there, if there's some target available in that specimen, then great. Do we have a drug in our arsenal that'll hit that target? Fantastic. That is precision, the current state of our world in precision medicine, right? It's finding a target that we know to exist in the tumor, and then hitting it with whatever we have in our arsenal. The problem is, in this case, is that what we've taken out of the brain, that specimen, does not necessarily represent the cells that are left behind in the brain. And so it's really the cells that are left behind that are the biggest drivers. And so we need to better understand what those cells are left behind, as well as the biology of what we have in our hands that have been removed from the patient's brain. So the biology is not as simple as one size fits all, the answer being red, but in fact, the ecosystem is quite complex. And I'm showing you 2 different shades of 2 colors, red and blue. And it's way more complex than that. It's definitely more like a rainbow. So it's definitely a complex, heterogeneous ecosystem, then worse yet, it evolves over time. So really the future of precision medicine is a better grappling or better understanding of the true extent of the heterogeneity across the entire patient's brain. And so that's where we started an effort a few years ago to collect aspects of that heterogeneity through image localized biopsies. So here's a patient that came in from an outside clinic that you see the edges of enhancement I'm showing you on different types of MRI are sort of segmented out. Red is what's T1 GAD, and T2 and FLAIR are kind of blue. And you see that this particular patient was seen in an outside clinic and then referred into Mayo Clinic for consideration for surgery. And this patient then showed up about 17 days after this image came to Mayo, and they showed up to the OR. And what you'll see is a pretty dramatic increase in growth of this tumor in those 17 days. So there's the expansion. So pretty significant growth rate in 17 days. And at the time of surgery, the patient received, I think, 11 biopsies. And you see them outlined on this image. And so by knowing the location of those biopsies relative to the imaging, I can then tell you, then we can sort of glue together our understanding of the heterogeneity within the patient's tumor and relate it to the patient imaging. So we've now done this for many hundreds of patients. Our consortium has access to, I don't know, something like 3,000 or 4,000 biopsies like this. Even in our cohort alone, we've collected almost exactly 1,000 biopsies at Mayo, Arizona, just in the last couple of years. So the idea here is that you're collecting these image localized biopsies at different locations. They're at whatever the surgical approach best supports for, and you might have these different 3 examples I'm showing you here. But you do have lots of different imaging features that you can characterize for each of these patients. You've got different types of MRI, different types of MRI that are related sort of loosely to different aspects of biology. So like T1 GAD MRI is distinctly related to leaking neovasculature. So it's the leaky angiogenesis, which allows for perfusion of these contrast agents. And so then you get a bright area. So there's tons of underlying biology, and there's certainly a ton of physics underlying these images. So one of the aspects of the lab that has been focused on the last several years is using these image localized biopsies in combination with these multiparametric MRIs, gluing them together and the feature sets associated with regional changes, with regional changes on the imaging to regional changes on the biopsies. And to allow that to use sort of machine learning or other artificial intelligence approaches to be able to build out predictive models of any aspect of biology we're particularly interested in. We have interest in lots of different aspects. I'll give you a couple examples here. So I'll go through a couple examples specifically, and as promised, we'll first talk about sex differences related to this. So if you're building these models, you've got to figure out make sure there's no biases in your data, right? It's one of the biggest problems in the world of radiomics, is that there's often biases in the data. So the first thing you look at, of course, is sex. And if you just look at the imaging correlates of prediction of the top 6 genes that are known to be aberrant in terms of like copy number in GBM, and these are those top 6 genes, you find that the imaging features that correlate with EGFR amplification in women are different than men. That's basically what this chord diagram says. So much so that, and the same thing's true for PDGFR, PTEN, you name it, there are different imaging features that predict that biology. Okay, interesting. So then when you apply this into a machine learning model, it turns out that if you pool all your data, which machines need lots of data to do a good job, because they've got to sample the diversity of all possible data, that's purple. The accuracy of those models are something like purple. If you separate out the male and female data, so you have lower N in each pile, but maybe closer to a single imaging set of features that are going to predict that biology, your accuracy tends to go up. So even though you're lowering the power of the model by decreasing the number of N, you're honing in on the signal more efficiently because the signals are just distinct from an imaging standpoint between males and females. Okay, so that's what this sort of summarizes. And so you can get reasonably accurate models, but what's the value of such of those models in predicting biology? So the value is exactly this model. So we've got a patient, pre-treatment imaging on the left, 1 month and 3 months after treatment. You see this sort of one region is kind of getting smaller, one region is getting bigger. This overall isn't changing all that dramatically. So is this patient responding? Well, this patient was receiving an EGFR targeted therapy. So wouldn't it be nice if we knew where the EGFR targeted region was? So this is a patient that was treated historically, and we applied a more recent EGFR amplified radiomics model to it. And what we find is the region where it was responding was the region where there was EGFR predicted to be amplified. So you expected drug has target, and target is there, and the combination created a response in that region. Whereas the region for tumor progression was outside of that region where we expected EGFR amplification to exist, based on the radiomics model. We've then done the same exact thing for a clinical trial that's focused on a variety of EGFR inhibitors. I'm just going to give you 3 patient examples. The top patients are ones that were the top is at the first treatment, the first imaging time point right before treatment, and then the second is 3 months in. So 3 months after receiving the first treatment, and you see that the patients that had their imaging abnormality grew was the patient that had no target present predicted by the radiomics model, whereas the patient that had the imaging target get much smaller was one where the drug was matched with the target. The target was present, and so there was expected to be a good thing. So you could imagine building clinical trials, and this is something we're working on, where you target and choose patients based on their imaging predictor of whether the target is present in any significant component of the disease. So the very last example I will give you on this front is the even more complex but even more urgently needed challenge of imaging changes and how it fuses to our understanding of changes in the immune response in brain cancer. So you think we don't know anything about whether EGFR is present or not in this disease. Now let's add in an entire system of the immune system and the immune response in the disease, and inside the brain where you have very little access to tissue, right? So it's not surprising that we just don't fully understand what's happening in individual patients. And so much so that this would be a typical course for a patient. And this has been the typical course for the last decade when EGFR, for which immunotherapies have been tried. So -1 month, patient comes in with what appears to be a recurrent glioblastoma. They confirm it 1 month later and say, yep, this thing is still growing, definitely need to start a new therapy. So they introduce the patient to an immunotherapy, 4 months in, still growing, 7 months in, still growing, wait, I think we're turning the corner, and then the lesion gets smaller and smaller. So this is the standard way that you see, this is the standard pattern that you would see across GBM patients with receiving immunotherapies. The problem is you can't tell at this point at 4 months and at 7 months in those early months, whether that is immune response or the tumor is actually responding to therapy. Because what I didn't tell you here is this patient never received a change in therapy. They just received the immunotherapy. So this early growth pattern here, what's called pseudoprogression, is associated with, is thought to be associated with a significant immune response. And there are some surgical studies that have proven that by going in, a surgeon, clinicians were very worried about this expansile behavior you're seeing here would go into a surgery in these early windows and then find that under the microscope, there weren't a lot of tumor cells. There was just this huge immune response. Okay, so distinguishing an immune response early is important because the sort of community level response assessment for neuro-oncology, IRANO, and I for immunotherapy, assessment point is 6 months. And what that means is a patient that comes in here with a recurrent GBM at day 0, they have a median survival of something like 4 to 6 months. The IRANO criteria says, okay, well, because we can't identify what imaging changes mean in those early months, do not change therapy again for this patient for 6 months. That's the general criteria, the overall or overarching theme of the criteria. So there's a disconnect, right? You're committing a patient to a treatment that you have no idea if you're going to find out if it works. And the only way you find out if it works is if they live longer than 6 months, basically. So it's a disconnect. It's a real pragmatic challenge, and we're trying to avoid waiting that 6 months to truly assess treatment response. So we partnered with one of these immunotherapy trials. We now have like 8 others that we're working our way through data-wise. And this was a dendritic cell vaccine. And a successful dendritic cell vaccine would take a patient receive pretreatment imaging, apply the vaccine early on, and then just track post-vaccine imaging. And a successful dendritic cell vaccine would be one in which T cells would be recruited. Basically, there'd be a significant T cell response. And there's not a ton of T cells in the brain, so maybe we can detect that with some imaging signal. So my MD PhD student that had just finished built exactly that. She basically took patient MRIs and built radiomics models for T cell prediction using our image localized biopsies. So she took all those image localized biopsies, she deconvolved what was T cell, and then used that to train these models. And when she did so, the resultant trained models she applied to the patients in the dendritic cell vaccine trial. Importantly, this is a sex-based radiomics model. So there's a male model and there's a female model. Those are distinct. And what you found is, I'll just give you two examples here, but I'll show you the whole cohort in just a second. Examples like this, you see patterns like this. We're on imaging at prior to treatment in 2 months, not 6 months, not 4 months, not later, 2 months, the very first imaging follow-up time point. You see that there's a decreasing pattern of T cells prediction, and the imaging is overall kind of shrinking, which is kind of not generally good in the immunotherapy context in those early days, right? So this patient ultimately lives five months with a decreasing pattern of T cells going down. The red disappears and it becomes what's called colder. Whereas the patient on the bottom has an increasing pattern of T cell patterning. It's not huge, but it's measurable. And what you find is this patient lives 37 months. You're like, okay, well, this is interesting. There are obviously different locations. There's lots of different heterogeneity. What if we just looked at those broad patterns across the patients? And this is what you see. Amongst the 20 patients in the clinical trial, those with increasing patterns had increasing patterns of more T cells do better, do significantly better compared to the decreasing patterns. And what's important is this clinical trial prior to this sort of analysis would be considered a failure, right? Because on average, the median survival didn't change all that much. There was no measurable huge win for this trial. And the PI of the trial was in fact working with a variety of us to figure out methods for, detecting, meaningful benefit in individual patients. And so this is basically what we found. So bringing this all together, sex matters, a heterogeneity of the disease matters, predicting our understanding of the overall disease dynamic matters, and that we can use the power of imaging, although it's an extremely challenging course because there's I can have a whole presentation on the challenges thereof. So the very last 30 seconds of my presentation, I wanted to mention everything I presented here had a significant focus on sex differences. And there are sex differences in omics biology really across the spectrum. The problem is we put in a sort of a call for action a couple months ago, well, it's now I think been a year, because it turns out the databases that you rely on when you do these omics analyses, whether it's transcriptomics, pathway analysis, you name it, whatever it is, generally are not curated for sex. So if there's sex biases in your data, if the sex biases exist, you have real problems because there's a disconnect between whether your male or female cohorts are going to be accurately represented on the other end. And so grappling with that is going to be a huge challenge for all of biology. So I have a cute little video I can send a link to that my daughter made, my 6th grade daughter made, to describe this disconnect. But you can reach out to me and I'll send you that. No, there you go. And thank you very much for your time.
Thank you so much, Kristin. It was awesome.
Should I stop sharing?
It's up to you in case you need some slides, but go ahead, Matt.
Oh, I don't.
Thanks, Kristin.
Never stop sharing, Kristin.
That was really cool. Jade, wait 1 second. I think I will try to catch some questions from the audience first. Any questions?
Oh yeah, no problem. I was just hanging that up. I'm ready whenever you are.
Jade, you're welcome to ask questions as well.
Hi, thank you for your talk. I was just wondering about the diffusion pattern in glioblastoma, or more like growth pattern, or maybe it's both. How does it happen that it spread so fast in that MRI? I mean, I understand it's probably because of the driver mutation as well, because it's aggressive by nature. But I'm also wondering if the sort of physiology of the brain, the way brain is built, somehow encourages faster diffusion. Thank you.
For sure, it's the architecture of the brain definitely allows for it. It's built, there's, definitely that's true. So there's definitely architecture, structural pathways that make this a possibility. The other is that, you think it from like a developmental biology perspective, there's lots of reasons why you would want nerve cells or nerve-associated cells to expand over long distances, right? So think of patterning within the embryo to build out the nervous system, period. And that patterning has to go over long distances, whereas in other organs, they're more discretely located, right? So the nervous system is an exception to that. So that's at least the general thought process, is that there's some aspect of this sort of tapping into the embryology of it all.
Sorry, can you hear me? Thanks for your talk. I guess I can't remember which model it was. I think the one that you were showing the driver gene predictions, but the one that you had the AUC results for. It seemed like even within those, for the split model, that a lot of times the female model predicted better. Do you know yet maybe what might explain that?
We don't, although it's been unbelievably fascinating. Basically, we see generally speaking, a little bit of a tighter signal there. Why? I don't know. I don't know. This is definitely something that we're way down the rabbit hole on trying to figure out.
Hi, Kristin, this is Ben. You probably didn't hear my talk before. I was going on for half an hour about ecDNA, extrachromosomal DNA. And there's now, this is very new, right? But there's strong evidence that in glioblastoma, this seems to be like a very, very important factor. And I was stunned by the sex differences that you showed, like the EGFR amplification and the P10 amplification in women, EGFR in men. I think we see these are often amplified, both of them are often amplified on on extrachromosomal DNA. So first question, can you see this in these diffusion patterns? Does it make a difference? And then, of course, that's a weird question, but why would there be these sex differences in terms of woman P10 being amplified on ecDNA and EGFR being amplified in men? And are there two classes of tumors, some driven by ecDNA and some not driven by ecDNA? Is there some evidence for that?
Yeah, so I guess starting with the first piece, why it would be different, there's tons of sort of evolutionary biology questions. You should corner Joel and ask him. He will tell you all the ideas why there might be sex differences. There's some really, really cool narratives about why there might be basic biological sex differences that drive the response to tumor and response to tumor insult. The patterning that was showing you the bar graph that was referred to by the previous question with regard to area under the curve, was the area under the curve was the accuracy of the model. And the model is just basically how closely does imaging glue to the biology? Like, does it predict the biology? And that's the distinction that's quite different, which is fascinating, that the imaging patterns are different. Now, why the biological choices we've made distinctly, the example that really got me was Josh Rubin's example 100 years ago when he first introduced me to the concept of even looking at this, was that in cyclical famines, as an example, just sort of zoom out, in cyclical famines pick anywhere in the world where there might be a cyclical famine, there's a normal number of female babies born, but they're abnormally small per pregnancy, whereas there's an abnormally few male babies born, but they're of typical size. And so if you have that cell sort of evolutionary driven cell decision making that says that we need a lot of uteruses relative to the number of males, then to maintain the overall population, you could imagine that a tumor would latch on to some aspect of that, right? And use that to whatever advantage it might need. And so you could see how there might be some different paths to how the biology might play out. And so I think they're relatively subtle because it's still the overarching system. There's tons of shared, right? But there's still going to be these just at the margins differences. And so why EGFR would behave that way, I don't know. But EGFR has some really interesting patterning differences, and PDGFR has some really interesting patterning differences. We're doing so with some experimental models right now. I feel like I'm really way over time, and I'm so sorry.
Do you have other questions? If not, then let's thank Kristin again.
Thank you guys so much.
Thank you so much.